Bismillah Rahman Rahim. We start the second video on question paper 2222 of November 23 exam. This paper is for 1 hour 45 minutes and it is for 80 marks. Now coming to question number 4, we've discussed the first 3 questions in the first video on this. Figure 4.1 shows a Siamese fighting fish that lives in fresh water, large colorful fin and location of an organ that can be filled with atmospheric air. Figure 4.2 shows the effect of changing pH on the solubility of oxygen in water. Now, whenever you look at a graph, always read the graph to yourself. How do you read it to yourself? On the x-axis, we have pH of water. So at 6 pH, we don't see any percentage solubility of oxygen in water. At this pH, we have something like less than about 58% of solubility. Now, as we are increasing the pH from 6.5 to 7, the solubility is increasing. You can see here it's increasing. It was here, now it's increasing. At 7.5, it is increased to 80%. And then at 8, it has increased to nearly 90. And then at 8.5, it has increased. And then around 8.6, it has increased to 100%. So here we have the percentage solubility of water. Of solubility of oxygen in water, we have the percentage solubility of oxygen in water, and as the pH is increased to more than 8.5, it has become 100% soluble in water. So oxygen, percentage solubility of oxygen in water. So read this very carefully. Here we have the pH of water, and then we are talking about a fish. Then figure 4.3 shows the effect of changing temperature on the solubility of oxygen in water. In the previous one, we were checking the pH of water, how it affects the solubility. Here it is talking about oxygen solubility in water in milligram dm cube. So we have oxygen solubility in water here, and here we have the temperature of water. Now in this situation, now something very different is happening. As the temperature of water is increasing from 0 to 5 to 10, 15, 25, 35 and 50, the oxygen solubility from 14 is going down to nearly 5.4. So the oxygen solubility, as the temperature is increasing, the oxygen solubility in water is decreasing. Here what was happening? As the pH of water was increasing, the solubility was increasing. Okay, let's look at the question now. When you've looked at the graphs very carefully, then you read the question and then again you go back to the graphs. Siamese fighting fish live in shallow water that can range in pH from 6.9 to 8.2 and in temperatures from 15 to 40 degrees Celsius. Use the information in figure 4.3 and 4.2. And what is 4.2? 4.2 is this one. When it says you refer to these, we have a lot of this in the A levels. Then you must look at these and just tell me something about these. So use the information of 4.2 and 4.3 to explain how the conditions in which these fish live may be challenging. Now let's look at it. How can we answer this question? So first of all, you are going to plot it on the graph. So here we have 15 degrees to 40 degrees. I have drawn these green lines. And you can see that at 40 degrees Celsius, the oxygen solubility is very low. So there's going to be less oxygen available for the fish. Let's look at the pH graph. The pH graph is between 6.9 to 8.2. 6.9 is the one which is the dangerous one because the oxygen solubility is low here. Here it is high, but here it is low. So at 6.9 we're going to have problems. So low oxygen availability for fish at the low 6.9 pH range and at the 40 degree temperature. So here and here. Why do we need oxygen? Less oxygen available for what? For aerobic respiration. And then you just tell me about this graph. What is happening as the pH is increasing, solubility of oxygen increases. So you said as pH increases, solubility of oxygen increases, which you can see here in the line. It is increasing. And then you said to me, as temperature increases, solubility of oxygen decreases. And that is what you can see from the graph. As temperature has increased from here to 50 degrees, the solubility of oxygen decreases. And then you gave me a data code. Data code means that you gave me some figures. And I'll give you an example of the figures at the end. Now, the wording is low oxygen availability for fish at pH 6.9 and 40 degrees of temperature range for aerobic respiration. And then you give me a general statement about the graph. As pH increases, solubility of oxygen increases. As temperature increases, solubility of oxygen decreases. So you're just telling me what you've seen in the graph. This is a very important exam skill. Please remember this. You just have to write what you see in the graph. And then when I said a data code, now I'm going to tell you what does the data code mean and what will you give me in the data code. In a data code, what you do is you take two extremes. I always say the beginning and the end. So I've taken 6.5 pH and 8.5 pH and I've given you the percentage solubility of oxygen in the water. So this would get you 1 mark. This is a data code. I said beginning and somewhere at the end. You could have the graph like this, then maybe you can give me a figure here, then you give me a peak here, and then you give me a figure here. But because this is a straight graph, so you take one of the beginning points and one of the end points. You can do any points. So this would be a data code for the pH. Now, a data code for the temperature would be any two temperatures. You could have said 5 and 50, you could have 5 and 45, you could have 10 and 40. So two datas, but the important thing is that you must give me the units. Here the unit was degrees Celsius and here the unit was milligram per dm cube. So at 10 degrees Celsius, 11.3 milligram per dm cube, at 50 degrees Celsius, 5.4 milligram per dm cube. So you must read off very correctly from the graph and give me a data code. This means a data code from any one of these would have got you 1 mark, either from the pH one or from the temperature one. Coming to question number part 2, all fish have gills that provide a gas exchange surface in water. Siamese fighting fish also have an organ that has similar features to those of a human lung. These fish swim to the surface of the water to take air from the atmosphere into this organ. Explain the advantages to Siamese fighting fish of also having this organ. Yeah, it would be useful when there is low oxygen or no oxygen. More gas exchange because gases will be exchanged with the air. More oxygen will be available for the fish for aerobic respiration to release energy, and the energy is needed for movement, for locomotion. So explain the advantage to the fighting fish of having this organ. So we'll just know how to word this. Then Siamese fighting fish have 42 chromosomes in each diploid cell. State each of the following. The name structure in each diploid cell that contains the chromosome nucleus. The type of nuclear division that produces gametes. Here is going to be where we are going to lose marks because we're going to write the spellings wrong. Meiosis. Then the number of chromosomes in each gamete. The number of chromosomes in each gamete would be 21. Why? Because it said to you here in the question 42 chromosomes. So that's the diploid number. So half of that would be 21 and that would get you your answer. Useful in no oxygen or less oxygen, gas exchange with the air, more oxygen available for the fish, for aerobic respiration, to release energy, and energy, you can give me any use of the energy. Energy for movement, energy for protein synthesis, energy for growth. And part C of the question, a very long question. The dramatic shape and color of Siamese fighting fish are produced by selective breeding. Suggest how the process of selective breeding in Siamese fighting fish can be used to produce these desirable features. Very easy. Selection is by humans. Selective breeding is another name for artificial selection. Selection is by humans. You choose the fish with the desirable features, then you cross them or you breed them, and then you have offsprings, or the next generation will be produced, and then the offspring with the best features will be selected again to breed, and this will be repeated over many generations. That's a typical answer that you would give for selective breeding or artificial selection. So, selection is by humans, choose fish with desirable features, then you cross them or you breed them, or you say the next generation is produced. So, and then out of the offsprings, you select the ones with the best features and you select them to breed them. And this is, of course, repeated over many generations. And this was a very, very narrow mark scheme. 5 marks, only 5 points. You had to give me all these 5 points to get your 5 out of 5. This is a total of 15 mark question, a very long question. Question number 5. A student investigated the use of an enzyme in fruit juice production. The student carries out the following steps. Cuts one apple into small pieces, puts the same number of pieces into each of 3 beakers, A, B, and C. Puts the same number of pieces. Now there's no this thing about the mass, but just cut into small pieces, and then put the same number of pieces in each of the 3 beakers. So we put in, say, 3 pieces here, so 3 pieces here, and 3 pieces here, and add 2 centimeter cube of a dilute enzyme solution to the apple in beaker A and B. So we've added a dilute enzyme solution in beakers A and B, and then we have added 2 centimeter of distilled water here. So here we have the enzyme, here we have the enzyme, a dilute enzyme solution, and in C we do not have any enzyme, we just have distilled water, and it's the same volume, 2 centimeter, 2 centimeter. Places each beaker in a water bath at a constant temperature for 30 minutes. So we place them all in a water bath for 30 minutes. And then filters the juice from each beaker into a separate measuring cylinder and records the volume of juice collected in each measuring cylinder. So we've cut the apple into small pieces. We put the same number of pieces into each of the 3 beakers A, B, and C. Then add 2 centimeter of dilute enzyme solution to the apple in beaker A and B. And then you add 2 centimeter of distilled water to the apple in beaker C. So apples plus the enzyme in A and B, and apple plus distilled water in C. Then you filter the juice from each beaker into a separate measuring cylinder and you record the volume of juice. Now it says identify the enzyme. The enzyme is pectinase, but if you had said cellulase, even that was okay. They accepted that. Pectinase. Then outline the reason why the student set up beaker C. Why? Because it's a control. And why do you set up controls? You set them up for comparison. To say, okay, did the enzyme make a difference, or was it just like that? Or how do you control it? How do you prove it that okay, the enzyme worked and then the enzyme did not work? So that's called a control. And basically the control is for comparison. The reason for a control is comparison. Then we come to the B part of the question. The result of the student's investigation are shown in table 5.1. Beaker A was kept at 45 and the volume of juice was 20 cm cube. B was 95 cm cube and C was 45 and 5 cm cube. Use your knowledge of enzyme action to explain these results. Now what has happened? Beaker A produced more juice than beaker B. B had less juice. Beaker A at 45 enzyme is working very effective collisions. Beaker B at 90 degrees enzyme has denatured, or you say the shape of the active site is lost. So in beaker B at 90 degrees the enzyme got denatured and the shape of the active site was lost and the substrate no longer fits. So we had to explain what? You had to explain the results of the investigation as shown in table 5.1. Use your knowledge, and this was another 5 marks. So we just word this. Beaker A produced more juice, beaker A enzyme working best, effective collisions, beaker B 90 degrees Celsius, so enzyme denatured, active site shape changes, substrate no longer fits. Or you can say substrate no longer complementary to the active site. Coming to the last part of this question, bacteria can be genetically modified to produce enzymes for use in biotechnology. State two reasons why bacteria are suitable for this purpose. Now, this is of course a syllabus point. If you look at it, this in the syllabus, there are no ethical concerns. Their presence of plasmids and bacteria reproduce very fast, or they have a very high growth rate, and then they divide by binary fission, and they're genetically identical. They are easy to grow, and they're able to make complex molecules. The exact points of the syllabus. So very quick recap, no ethical concerns. If we do these things on animals, and we would have, okay, what are we, it's wrong. So no ethical concerns, their presence of plasmids, rapid growth rate, and they divide by binary fission, so they're genetically identical. Then it's very easy to grow them on petri dishes. They don't require so very complex labs or farms. Able to make complex molecules. Bacteria can make complex molecules, so that's why they are used in biotechnology. Then coming to question number 6, figure 6.1 is a cross section of a plant leaf. You can see J, it's a cuticle, it can allow light to pass through. K is the palisade mesophyll, it contains chloroplast, so absorbs light. L is the lower epidermis, which contains the guard cells and the stomata, and that is for the carbon dioxide in and oxygen out. So you were supposed to identify each of J, K, and L and explain the importance of each in the process of photosynthesis. You see, you could have said cuticle and it protects it from pathogens, but they didn't ask you that question. They said outline and explain the importance in the process of photosynthesis. They didn't say outline its importance to the plant. So the cuticle also protects the leaf from invading pathogens, just like we have skin, which protects us from invading pathogens. Then K was the palisade. Then K were the palisade mesophyll cells, and they have chloroplasts, or you can say chlorophyll. So the chloroplasts contain chlorophyll, and that absorbs light. L was the lower epidermis, and it has the guard cells, or you could have said stomata, and they allow the carbon dioxide into the leaf, and they allow the oxygen to move out of the leaf or diffuse out of the leaf. The B part says name and describe the function of one tissue found in the vascular bundle of a leaf. Well, you could have talked of the xylem, or you could have talked of the phloem. I'm not saying both. So you could have talked of the xylem or the phloem. If you talked about the xylem, then its transpiration transports water and mineral ions in one direction, and it supports the lignin in the xylem supports the plant. If you talk to the phloem, then in translocation transports sucrose and amino acids. It can be up or down, and it is source to sink. So you could have given me either the xylem or the phloem, and these are points, and I was just doing a June 23 paper, and the same things have come in that as well. So these are the very common topics that are going to come in all these papers. So phloem, translocation, transport sucrose, or you could have said amino acids, up or down, and source to sink. Xylem is transpiration, transports water and ions in one direction only upwards and supports the leaf. Now coming to the last question, mangrove trees grow in forests that form an important coastal ecosystem. Mangrove forests have a high biodiversity with many different animal species found in them. Describe what is meant by each of the following terms. Ecosystem, living component, 1 mark, non-living component, and interacting with the living component, 2 mark. Then it says biodiversity, number of different species in an area. Number of different species in an area. Species, group of organisms that can reproduce to produce fertile offsprings. So basically these definitions are all there, which were direct questions. In species, you have to say group of organisms that can reproduce and then to produce fertile offspring. So that got you your 2 marks. So living component means the fish, the trees, the plants, and the non-living component means the water, the soil, the air, and all these interacting with each other. Like if there's water in a pond, then there'll be fish in the pond. If there's no water, there'll be no fish, they'll die. Biodiversity means number of different species in an area. The number of plants, more the number of plants, more the biodiversity. More the plants means more the birds. More the birds means more the insects. more butterflies. So number of different species in an area. Species, a group of organisms that can reproduce, to produce fertile offspring. I'll just give you a detailed definition of species at the end. So, definition of a species is a group of organisms that may interbreed and produce offspring that is also capable of producing the same kind. Or we can say, what is a species? Remember, a species is a group of organisms that can breed and produce viable and fertile offsprings. Two animals are not the same species if they cannot mate and produce a living fertile baby. So coming to the last part of the question, B, like many forests around the world, mangrove forests are vulnerable to the effects of human activity. Explain how forests can be conserved as a sustainable resource. Education, or you can raise the awareness, then there should be laws which are in which there are quotas that you can only cut down so many trees in a year, or you can have the protected areas and you can monitoring that how many trees should be cut down because we're talking of mangrove forests. And then we take measures to reduce or stop the deforestation or cutting down of trees. Then trees are cut only from selected areas, and then you do reforestation or replanting so that the trees are replaced, and then we produce so that they can be produced as rapidly as removed. So raising awareness among the people and educating them so that people don't, we don't cut down trees, and we take alternative strategies, and we do not like recycle paper. So raising awareness, then quotas or laws to protect the areas, then stopping deforestation, then trees cut from selected areas only, then replanting, or we can say reforesting, and then so that they are produced as rapidly as removed. Now, another word that has been used in the mark scheme is coppicing. Now, I want to talk about coppicing. What is coppicing? The tree to be coppiced is cut close to the base in winter, and the shoots rapidly regrow from the stool the following spring. Coppice ready for harvest between 7 to 20 years. So, this is called the coppice cycle. And this can also be done to ensure that, they're used, and then, of course, they regrow in 7 to 20 years. And so the numbers will remain the same. Thank you very much, and I'm giving you all my mobile number in case you have any questions. You can WhatsApp me, and I will try to answer as best as I can. If you have any questions regarding the syllabus, or if there's anything that you want to understand, so you can send me a picture of that question, and I will send you a voice message, or somehow I'll explain it to you. Thank you very much, and we will continue with the rest of the paper 4s in the next couple of videos.